environment. So efficient cloning of organisms. It may be developing a large number of individuals within a short span of time. Plant biotechnology. Plant biotechnology. Okay, so these are certain applications with respect to biotechnology. There are many more. We have marine biotechnology. So when you talk about medicine, so genetically engineered vaccines. So we have generally recently we faced with a pandemic situation where many individuals were affected by a small virus, but how to control that virus was a big question, and we introduced genetically engineered vaccine. Okay, so genetically engineered vaccine, it may be uh, a recent or earlierly developed vaccine. We have humulene, which is also called as human insulin. Okay, so ಇದನ್ನೆಲ್ಲ ನಾವು ಮೆಡಿಸಿನ್ ಮುಖಾಂತರ ಅಥವಾ ಬಯೋಟೆಕ್ನಾಲಜಿ ಇನ್ ದ ಫೀಲ್ಡ್ ಆಫ್ ಮೆಡಿಸಿನ್ ಮುಖಾಂತರ ಪಡ್ಕೊಂಡಿದ್ದೀವಿ. ದಟ್ ಈಸ್ DNA recombinant technology was followed. Then we have certain antibodies which were generated. We call it as monoclonal antibodies using it is used in diagnosis of certain diseases, or it may be used in curing certain diseases like cancer or uh, uh, sexually transmitted diseases, etc. Gene therapy. Gene therapy is again related to medical biotechnology, which talks about a treatment of diseases like cystic fibrosis, or it may be Hutchinson disease, or it may be ADA disease, that is adenosine deaminase deficiency disease. So all these are application with respect to biotechnology in field of medicine. Then industrial biotechnology, we gain different products of produces like ethyl alcohol, or it may be lactic acid, citric acid, glutamic acid, basic amino acids which are derived from species of fungi or bacteria, right? So ಮುಂಚೆ ನಾವು ಆನಿಮಲ್ ಸೋರ್ಸ್ ಇಂದ ಕೆಲವೊಂದು ಅಮಿನೋ ಆಸಿಡ್ಸ್ ಅನ್ನ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡ್ತಿದ್ವಿ. ನಾವು we are introducing the genetic material in the bacteria and place them in the environment where they are producing these amino acids, and we are utilizing them, which is totally safe. Why I call it as safe? Because the producers which are derived from some other species, some other group of organism may develop allergy in certain period of time, after certain period of time of their usage. That is why enzymes, okay, we use basic enzymes like lipases, or it may be proteases. Okay, so all these are extracted from bacteria, okay, or a strain of fungi. So we also use single cell protein, and this is purely fungi, right? So single cell protein from fungi like mushroom is very best example. Or we have another species, that is methylotrophus. Methylotrophus, which is a bacteria used in extraction or production of protein in larger concentration. Then animal husbandry, now basic methods and follow ಮಾಡ್ತೀವಿ, like MOET, that is multiple ovulation embryo transfer, or gene transfer, okay, or it may be recombinant DNA, rDNA technology, where we develop genetic material which can produce protein, and that protein is essential for our body. And who is going to synthesize it? That is animal. We introduce a DNA, that is recombinant DNA in animal will produce protein, and that is essential for our body, and we consume it. So mammals ಇಂದ ಬರುವಂತ ಮಿಲ್ಕ್ ಈಗ ನಾವು ಕ್ಯಾಟಲ್ಸ್ ಇಂದ ಬರೋ ತರ ಮಾಡಬಹುದು. If any mother is failing to lactate, which cannot produce milk after parturition, and baby will be deficient of nutrient, so the cattle's milk will be a supplement for that newborn individual. So that is again an achievement through biotechnology. Okay, so environmental biotechnology, it is using a bacterial strains efficiently. We use Agrobacterium tumefaciens, that is tumor-inducing plasmid, TI plasmid, is extracted from this agrobacterium, and we are using that particular feature in developing roots, hairy root, or it may be developing multiple shoot in a plant, so that they are having better yield or benefit. Apart from developing hairy roots by TI plasmid, we also use some microorganisms in micro preparation with respect to tissue culture. So tissue culture is basically a method where within a short span of time we are going to develop large number of offsprings and large number of individuals. So again, with the help of uh, tissue culture or your cryopreservation or uh, micropropagation, we are going to develop more number of individuals in small time. Germplasm. So in previous class, I just defined what is germplasm, right? So germplasm is collection of all possible genotypes, or collection of all possible genetic material within the organism, and that can be preserved by cryopreservation. Cryopreservation. So what is this cryopreservation? Placing the organism or organ at minus 196 degrees centigrade, which is on temperature of liquid nitrogen. Okay, so that is how cryopreservation can be followed in preserving germplasm, as well as preserving gametes, or preserving embryonal structure for longer duration or longer period. ಅರ್ಥ ಆಗ್ತಾ ಇದೆಯಾ? ಸೊ ಇದು ಬೇಸಿಕ್ ಇತ್ತು. ಬಯೋಟೆಕ್ನಾಲಜಿ ಯಾಕೆ ಕಲಿಬೇಕು, ಯಾಕೆ ಅದನ್ನ ಅಪ್ಲೈ ಮಾಡ್ತಾ ಇದೀವಿ, ವೈ ಆರ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಅಪ್ಲೈ ಇಟ್, ಹೌ ಇಟ್ ಈಸ್ helpful for our to our mankind. Okay, so in the same way, strategies in enhancement of food production talked about, or it spoke about development of resistant varieties. So biotechnology, genetic engineering, or bioprospecting will talk about, or it generally focus on how to improve crop varieties, how to improve genetic variability within the organism, so that we are getting benefited. So the basic tool which is meant for transferring gene or genetic character is plasmid. Okay, so plasmid is an extracircular DNA which is present in prokaryotic organisms. So if you take this as a prokaryotic organism, and if there is an extracircular DNA, so this extracircular DNA is called as plasmid. Okay, it will have its own genome, perhaps, but plasmid will contain certain factors. So if you observe plasmid, each plasmid will have origin of replication. If you take PBR plasmid, PBR, that is 322 plasmid. So adರಲ್ಲಿ it will have different portions altogether. What are the different portions associated with? We have ampicillin resistant gene, so AMP resistant gene. Then it will have tetracycline resistant gene. Tetracycline, it is TET resistant gene. And the portion where replication originates, and the ORI, origin of replication. Okay, so all these are, say, representing segments within a plasmid. So what is the significance of these structures? When you talk about ampicillin resistant, that means the bacteria which is having ampicillin resistant, they can grow in presence of an antibiotic, that is ampicillin. Hagene, if the bacteria is having tetracycline resistant gene, it can grow in the presence of an antibiotic, that is tetracycline. So these will inevitably be used as a selectable markers. Okay, so these markers can help us in identifying whether the transformation has taken place or not. So the basic tool for for biotechnology or genetic engineering are our enzymes, that is called as the restriction enzymes. Okay, so restriction enzymes were first observed in prokaryotic organisms. They were first seen in prokaryotes. And these prokaryotic organisms, say bacteria, used restriction enzymes to avoid infection or infestation by bacteriophages. Okay, so now we are using them as a tool. So there are restriction enzymes are used to cut DNA at particular position. They are used to cut DNA at a particular position. If the restriction enzyme cuts DNA at external side, external or periphery, we call it as exonucleases. That means it will cut the DNA at peripheral end or external end, somewhere like this. Okay, it will be cutting the DNA in this position. That is called as exonucleases. If the restriction in nuclease enzyme cut the DNA in center, okay, in between the peripheral region, we call it as endonucleases. That means it will cut the DNA at a particular position within somewhere here. So they are called as endonucleases. So what is the use of detaching the DNA? So if we have a parental DNA, for example, this is parental DNA, okay, and I have a genome somewhere like this, and I want a fragment of the DNA to be detached, like this. Okay, fragment of DNA to be detached like this, and I want to shift them. Okay, let me take it here. So this is parental DNA, and I want to shift the parental DNA or modify them. So this, I will take it out. And I will introduce the foreign DNA like this. Okay, so this is parent A and this is parental B. Okay, what I did, I introduced parent B gene in A. So now this will be called as hybrid. Why did I do that? So that the parent A is producing necessary character. So this is called as your development of recombinant DNA. Okay, introducing a foreign DNA into the parental DNA. So if the parental DNA is a plasmid, say parental DNA is a plasmid, and a foreign DNA is a linear DNA. If I separate the portion from linear DNA and separate a portion from parental DNA, and if I or your plasmid and introduce the parental DNA into your plasmid, then this plasmid will be called as recombinant DNA. ಏನಂತ ಕರೀತಾರನ್ನ recombinant DNA ಅಂತ ಕರೀತಾರೆ. ಸೊ ಯಾವಾಗ ಈ ಪ್ಲಾಸ್ಮಿಡ್ ಮಲ್ಟಿಪ್ಲೈ ಆಗುತ್ತೋ ಈ ಫಾರೆನ್ ಜೀನ್ ಕೂಡ ಮಲ್ಟಿಪ್ಲೈ ಆಗುತ್ತೆ ಆರ್ DNA ಕೂಡ ಮಲ್ಟಿಪ್ಲೈ ಆಗುತ್ತೆ. ಸೊ ಯಾವಾಗ ಈ ಪ್ಲಾಸ್ಮಿಡ್ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಮಾಡುತ್ತೋ ಅವಾಗ ಈ ಫಾರೆನ್ ಜೀನ್ ಕೂಡ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಮಾಡುತ್ತೆ. ಆಯ್ತಾ? ಇದನ್ನ ನಾವು recombinant DNA technology ಅಂತ ಹೇಳ್ತೀವಿ. If I take a plasmid, one example, last example, if I take a plasmid and introduce a DNA portion of DNA that is producing insulin. So this is producing insulin. Okay, I introduced a plasmid with a gene that produces insulin. So when this plasmid multiplies or expresses protein, all the time it is going to produce insulin. Okay, so that is how your bacteria is now involved. Bacteria is now involved in production of insulin. Okay, and we extract it and sell it in market. So that is again a benefit of recombinant DNA technology. Recombinant DNA technology. So ಈ restriction enzymes ಅನ್ನ ಈ ಆಡು ಭಾಷೆಯಲ್ಲಿ ಕಾಮನ್ ಲ್ಯಾಂಗ್ವೇಜ್ ಅಲ್ಲಿ we call it as molecular seizures. Okay, why it is called as molecular seizures? Because they are used to cut the DNA portions at a particular end, and they always cut the DNA wherever we or wherever the DNA is having palindrome. Palindrome ಅಂದ್ರೆ when you read it from right hand side or when you read it from left hand side, it should be the same. For example, M A L A Y A L A M. Okay, so when you read it from the left hand side, okay, from this side, it will have the same alphabets. When you read it from the right hand side, if I read it from this hand side, again it will have same alphabets, isn't it? So such sequences of DNA, we call it as palindromic sequence. Palindromic sequence. For example, G A A T T C G A A T T C. So this will represent palindromic sequence of that DNA. And your endonucleases or exonucleases will cut the DNA at this portion. So that is where your exonuclease or restriction endonucleases or restriction exonucleases will work. Now, yes, we have cut the DNA. Now we have to join the DNA. Who is going to join the DNA? Joining DNA is done by an enzyme called as ligase. So we observe this particular enzyme in a particular species of virus like HIV. So what it will do, virus, when it introduces double-stranded RNA in our body, with that it also has reverse transcriptase enzyme. By that, it will reverse transcribe and produce viral DNA, right? And this viral DNA will be ligated to, will be attached to host body. Okay, it will be attached to host DNA. Let us presume this is host DNA. So when it is attached to host DNA, that mechanism is done by an enzyme. We call it as DNA ligase enzyme. DNA ligase enzyme, right? So that is how your DNA, foreign DNA, will be introduced into a parental DNA, or it is ligated there. Perhaps it is having palindromic sequence in them as well. So just we are going to replace a portion of DNA segment within the plasmid, and that plasmid will be given into the organism. Okay, so identification of diseases, identification of factors are done with the help of biotechnology, and we have application or terms which are used. Example, enzyme linked immunosorbent assay or Western blotting. Western blottings. So these are methods used in identification of human immunovirus or acquired immunodeficiency syndrome diseases. Okay, so this was with respect to principles and process, what we learn or what we are going to define. In application, we will be learning about genetically, or you learned about genetically modified organism. So in that, you saw that agrobacterium or Bacillus thuringiensis, Bacillus thuringiensis, Bacillus thuringiensis contain a plasmid, and that plasmid contain cry gene. So cry gene, there are different types of genes. That is cry1AC, cry2AB, and cry1AB. Okay, so cry1AB, cry1AC, cry2AB, and cry are the three different cry proteins or cry genes. So what these cry genes do, they help in controlling different insects. For example, cry1AC, cry2AB will control cotton bollworm. Okay, whereas cry1AB controls corn borers. ಏನದು ಅಂತ ಅಂದ್ರೆ when we have say plant, and when we introduce that plant, say this is leaf, and I introduce that plant with cry gene. Okay, so if any insect, say this is the insect, if any insect come and feed on that plant part which was introduced with cry gene, okay, or which is associated with cry gene, ಅವಾಗ ಏನಾಗುತ್ತೆ, insect has an elongated alimentary canal, right? It is having tubular alimentary canal. So this is tubular alimentary canal. Okay, so cry protein will enter, or cry gene will first enter in that body. Cry gene will enter in that gut canal, and it will start producing proteins. That protein will slowly attach to the intestine, and it will rupture the gut canal of that insect. Okay, so insect which is feeding on plant. So this is plant introduced with cry gene. Okay, once it feeds on that plant part, cry gene will enter in the gut canal, and it will rupture the gut. How it is going to rupture? Cry gene will produce cry proteins. Okay, that is mRNA and protein. So cry protein are toxic proteins. Okay, they create perforation or holes in the gut canal, and it will kill that insect. Now the question is, if this is associated, say, with bt brinjal or cotton brinjal, if it is consumed by human, what may happen? Whether cry proteins are going to express or not? No. Why? Because this cry proteins only express in alkaline condition, and human gut will have HCl, that is acidic, and hence cry proteins get denatured in human gut, and it expresses only in alkaline condition. To produce cry proteins, that is toxic proteins, it kills by creating perforation or holes in that individual. So that is another point. Apart from developing insect resistant varieties, we also have your pest resistant. Okay, so I will stop after this and we will do some MCQs. For example, this is a root of your tobacco plant. Okay, so in tobacco plant, we introduce RNA, which is complementary to the RNA developed by nematode. Okay, so what is this all about? Nematodes are the smaller worms. Okay, so nematodes are the smaller worms. What they do is they introduce RNA into the root and modify it in such a way that they start producing protein which is required for the nematode. So now nematode will feed on the protein. If nematode is feeding on protein, plant will be deficient of protein, right? And it will slowly die. In order to avoid this, what we do, we introduce complementary RNAs in a plasmid. So now when nematode is producing RNA, or introducing an RNA, whatever RNA we introduced, it will go and develop complementary strands with the nematodal RNA. Okay, it will develop a complementary strand. So now this will act as double-stranded RNA, and double-stranded RNA will not express. Okay, if it is not expressing, there is no protein. If it is not producing protein, nematode will starve. Okay, so that is what all about RNA interference. We call it as pest-resistant variety. Okay, so because tobacco is an important cash crop, generally we develop these varieties in order to avoid nematodal infection or reduction of yield. Yes. Yes,
sir. Hamsana wanted to ask.
Yeah, go ahead. Yes, Hamsana, go ahead. You can type or you can unmute yourself. Yes, Hamsana. You raised your hand.
Hamsana, you are allowed to talk.
You can unmute and talk. Okay, so let us enter into MCQs.
I think Hamsana, she unmuted, she muted and not able to ask the questions. You can continue MCQ, sir.
Yeah. Yeah, biotechnology. So hope you are viewing the questions. Question number 1. In a recombinant DNA, a recombinant DNA molecule is produced by joining together an mRNA with a DNA segment, mRNA with a tRNA segment, a tRNA or two mRNA or two DNA segments. What is recombinant DNA molecule? Whether it is option 1, 2, 3, or 4. Pest resistant, yeah. Pest resistant, where we have RNA interference technology. In RNA interference technology, we introduce a complementary RNA strand, that is mRNA strand, to
Sir,
the
sorry to
RNA which
interrupt you,
was.
sir.
Yes.
Sir, sorry to interrupt. That pest resistance, they want repetition.
Yeah, I'm doing that. So, yeah. So if this is say root of tobacco plant. Okay, what is the role of a root? It will store nutrient, basically protein. And protein is a result of RNA translation, right? But nematode is a worm which will be infesting plant and utilizing it for protein synthesis, because nematode cannot synthesize specific protein. So what it will do, it will introduce RNA in the roots of tobacco plant, and now this will express or produce protein. Okay, so that will be fed by nematode, and nematode will survive. So that will be a pest. Okay, so what we did, if this is RNA strand introduced, okay, we develop a complementary strand for that RNA, because we already know which is nematodal RNA and what are its components. So what we did, we introduced a complementary strand to it. So what is the function of complementary strand? It will just go and align around it or beside it. Okay, beside it. So when RNA is single stranded, okay, when RNA is single stranded, it will translate and produce protein. When RNA becomes double stranded, in case of RNA interference, there is a double stranded RNA because we have introduced a complementary strand. So this RNA will not be identified by tRNA. It will not be identified by ribosomes, and hence it will fail to produce fail to produce protein. And this is also called as silencing of mRNA, silencing of mRNA by nematode or of nematode. Is it? So if RNA is not synthesizing protein, okay, your nematode will not get nutrient. Okay, then the plant will be resistant to nematode, resistant to nematode. So how we did that? We took an plasmid. Okay, we developed a DNA fragment, DNA fragment. Okay, which is complementary or which is producing complementary RNA strand, and that complementary RNA strand will be aligned to nematodal RNA, nematodal RNA. Okay, and it will become double-stranded RNA, which fail to produce protein. So this effort was done by the scientist to avoid infestation of a nematode. Name of the nematode is Mcqs incognitia. Okay, Mcqs incognitia is the name of the nematode. Okay, so nematodal RNA is silenced by the complementary RNA which is introduced or which is synthesized. Who is going to synthesize it? Plasmid. And who is going to introduce that plasmid? Us. We are going to introduce that plasmid. Is that clear? Okay, so it happens, everything happens under the soil. And now nematode will fail to gain nutrition. Okay, so we'll go back to MCQ. Yeah. Recombinant DNA is produced by joining two DNA segments. Is that clear? Ah, a gene produced for recombinant DNA technology contain a gene from an organism joined to a regulatory sequence of another gene. Such gene is called as oncogene, junk gene, chimeric gene, none. Okay, a gene produced for recombinant DNA technology contain a gene from organism. Andre, if I have a plasmid, if I have a plasmid, so this is the plasmid. I have a plasmid. Okay, plasmid have its own DNA sequence, and there is a DNA from other animal. Say this is a DNA from other animal. I want to separate a portion of this DNA, and introduce it in plasmid. What is this DNA called as? The portion of DNA is called as foreign DNA, but scientifically, that foreign DNA is called as chimeric gene. It is called as chimeric gene. Junk gene in the sense it is a waste gene. Okay, oncogene is the gene which is coming from a virus. Okay, I will just throw some light on this. Oncogene, oncogene is a viral gene. So this is chimeric gene. Oncogene, oncogene is a viral DNA fragment that cause cancer. So cancer causing viruses, we call them as oncogenic virus, and they contain a gene called as C-onc gene. Okay, hope you will remember this. Oncogenic viruses are the one which causes cancer. Okay, and C-onc are the gene which is associated with it. Is that clear? Is that clear? Okay, shall we move on to the next question? That is, a group of genetically similar organisms obtained from sexual reproduction is called as genetically similar organism obtained by, it is asexual, right? It is asexual reproduction. There is a gap between. So it is asexual reproduction. What they are called as? Whether it is clone, population, assembly, none. They are called as clones, right? They are called as clones. So individual unit of clone is called as ramete. Ramete. Ramete is individual. Clone is a population. Say similar organism, when they are placed together, we call it as clone. So individual organism, this organism, it will be called as ramete. Make a note of it. Then, to be useful in preparation of recombinant DNA, a plasmid must have no origin of replication and origin of replication, the ability to alternate between a linear and circular forms, restriction endonuclease activity. Restriction to endonuclease activity, ability to alternate between linear and circular forms, and origin of replication, no origin of replication. Yes, exactly, Ankit. It is origin of replication. DNA or plasmid should contain ORI, that is origin of replication. Very good, very good, very good, Tejaswini. It is origin of replication. So what happens at origin of replication? The primers or say RNA polymerase enzyme will go and bind there, and that is a site where replication will initiate. Replication will initiate. Then restriction endonuclease have ability to cut DNA at random site, at a specific site, both A and B, DNA and RNA at random site. Fifth, restriction endonuclease, whether it cut DNA at random site and what do you mean by random or sorry what do you mean by a specific site? It will cut the DNA either it will cut the DNA at the periphery, okay, or it will cut the DNA at the center, isn't it? Very specific. So it will be only observing palindrome sequence. If they are read from either side, it will be of same nature. 5 prime to 3 prime or 3 prime to 5 prime. If you read, it will be of same nature. Such DNA sequence, we call it as palindromes. And DNA will be cut at the palindromic site, at this specific site, right? Endonucleases, a group of enzymes which cleave DNA externally, internally, both 1 and 2, nor A nor B. Endonuclease. Endo andre ಏನು? Internally. Remember. Okay, it will cut the DNA at the internal portion or segment. Now, the extra chromosomal, self-replicating, double-stranded, closed circular DNA molecule are called as plasmid, phages, viruses, chloroplast. What it is called as? Right? Extra chromosomal, circular DNA. What it is called as? Extra chromosomal circular DNA. Plasmid. Extra chromosomal circular DNA is called as plasmid. Remember this. Huh? It is called as plasmid. They are not phages, it is not virus, it is not chloroplast. Plasmid consisting of its own DNA, okay, consisting of its own DNA with a foreign DNA inserted into it. It is called as. What is a plasmid introduced with foreign DNA called as? What is a foreign DNA introduced in a plasmid called as? It is called as recombinant DNA, right? It is called as recombinant DNA. Plasmid consisting of its consisting of its own DNA with a foreign DNA. Okay, a plasmid introduced with a foreign DNA. It is called as rDNA or recombinant DNA. Someone told it. Very good. Insulin, a protein consisting of 2 polypeptide chain, 3 polypeptide chain, 4 polypeptide chain, more than 4 polypeptide chain. What is insulin? Whether it has 2 polypeptide chain, 4 polypeptide chain, more than 4 polypeptide chain, or 3 polypeptide chain. Let me explain this part. Insulin, insulin, insulin is a hormone, or sorry, it is an enzyme, okay, which controls glucose metabolism. Okay, insulin controls glucose metabolism. This insulin is produced by produced by pancreas, right? And there are two portions of insulin. One, though, we call it as A peptide. A peptide. Then it has a B peptide. B peptide. But A peptide and B peptide are linked together by a disulfide bond like this. It is linked together by a disulfide bond. So there will be disulfide bonds here like this. Okay. So this is activated insulin. But we don't produce this activated insulin. We get proinsulin. Proinsulin. Okay. So this proinsulin will have A peptide. It will have a B peptide. It will have a B peptide. Okay, A peptide, and say this is B peptide, and this is A peptide. A peptide. It is linked together. It is linked together by C peptide like this. So this is C peptide. C peptide. This is, say, B peptide. B peptide. And this is A peptide. Right? So this is called as proinsulin or inactive insulin. Inactive insulin. But at the time of activation, so there will be disulfide bond like this. They are linked again by disulfide bond like this. But during activation, your C peptide will get separated. C peptide bond will get separated. And both A peptide and B peptide will be linked. So A peptide and B peptide will be linked. So that is how we get activated insulin. Activated insulin. But he is asking how many peptide bonds are found in insulin. That will be A peptide and B peptide. How many? 2 peptide bonds will be found. So another interesting fact is A peptide ಏನಿದೆಯಲ್ಲ, it is developed by a particular DNA fragment. It is produced by a DNA fragment. So this amino acid is produced by a particular DNA fragment. ಹಾಗೇನೆ B peptide is developed by a particular DNA fragment. DNA fragment. So what we do is we introduce this DNA fragment in a bacterial cell. Introduce the DNA fragment in bacterial cell. Say this is the DNA fragment which produces A peptide. And in the same way, we introduce the DNA. Same way, we introduce the DNA fragment which produces, say, B peptide in a particular DNA. So now, when DNA is placed in glucose solution, they start producing A peptide chain, and they start expressing B peptide chain. And now we extract both the A peptide and B peptide chains from the bacterial culture, and we develop disulfide bonds. Disulfide bond. So this will be called as insulin, which is artificially synthesized. Okay, why we are going to synthesize this artificially? In order to fulfill the demand of the population suffering from diabetes, right? Isn't it? So that is that simple. If a bacteria is going to synthesize insulin, we are not depending on other sources. So before recombinant DNA technology, the polypeptide for insulin was extracted from swines, that is pig liver, pancreas of pig. And people started to develop allergy with that. In order to avoid that, scientists gave an innovative idea where we are now depending on rDNA technology, rDNA technology for production of insulin, and this is called as humulene, which was developed by a company called as Lilly Company. Eli Lilly Company, right? You know this fact, right? So the next question which we are observing here, insulin, it contains how many polypeptide chain? It consists of two polypeptide chain. Got it? Why it is having two polypeptide chain? Because it has A chain and A polypeptide chain and B polypeptide chain linked by disulfide bond. Is it? Then the first human protein produced through rDNA technology was insulin, erythropoietin, interferon, somatostatin. 10th. It was insulin. Very good. Very good. It is insulin. So insulin was the first human protein through rDNA technology. Humulene, a genetically engineered insulin, was produced by which company? Just now I told you, whether it is Biocon, Glaxo, Eli Lilly, or Cipla. Which company, which company helped in evolving this particular product, that is insulin? It was, yes, exactly, very good. It is Eli Lilly Company. So Eli Lilly Company evolved it for the first time. The first licensed drug produced through genetically engineering. Licensed drug produced through genetic engineering is hmm. Can anyone tell which is the first licensed drug developed through genetic engineering? Again, it is humulene, that is insulin. Insulin was the first licensed drug developed through genetic engineering. Very good. Insulin, not penicillin. Penicillin, we didn't apply genetic engineering. Why? Because penicillin, before genetic engineering, it was used in during World War. During World War. So directly the spores were used, or directly the fungi was used, or in some reference book they say that where it was grown, say it was grown in on a piece of bread, or if it is growing on piece of waste material, that itself was used as an antidote for the bacterial infection. Okay, so bread ಅನ್ನ ತಗೊಂಡು ಮೈಗೆ ಇಡ್ತಿದ್ರು, ಅಥವಾ ರಾಟನ್ ಫ್ರೂಟ್ಸ್ ಅನ್ನ ತಗೊಂಡು ಮೈಗೆ ಇಡ್ತಿದ್ರು ಅವಾಗ, ಬಿಫೋರ್ advancement in medical science. Can you name the scientist who gave penicillin? Quickly, can you name the scientist who gave penicillin? Name the scientist who gave penicillin. Alexander Fleming. Very good. Yes, Priyanka. Yes, Pawan. Yes, it is Alexander Fleming who gave penicillin. Okay. Next. Before production of recombinant insulin, insulin for the treatment of diabetes was extracted from healthy humans, dead human body, dogs and cats, cows and pigs. 13th question. Before production of recombinant insulin from for the treatment of diabetes in human was obtained from, where it was obtained from? where it was obtained from. Yes, where it was obtained from. It was obtained from cows and pigs. Very good, very good. It was obtained from cows and pigs. Next, the plasmid generally used in production of recombinant insulin is, okay, plasmid used in production. This is not there in NCRT. Plasmid used in production of recombinant insulin is, yeah. Yes, can anyone?
Sir, two students answered as TI plasmid.
It is not TI plasmid. Hmm. So specific is PUC 18, but some references say that it is TI. You can go for TI plasmid. As it is not there with NCRT, we can rely on TI plasmid. Why? Because TI plasmid is explained, and most of the genetic engineering work is included with TI plasmid. Okay, so TI plasmid can be considered as an option for this particular answer or question, right? So plasmid generally used for production of recombinant insulin or used in recombinant DNA technology is TI plasmid. Then in one of the technique of recombinant insulin production, the gene of alpha and beta, that is A and B polypeptides, were inserted in the plasmid by the side of ORI, antibiotic resistant beta galactoside gene or restriction. Okay, it was introduced by the side of beta galactosidase gene. So if you take beta, this is the bacterial cell. Okay, let us presume this is bacterial cell, and bacterial cell has a plasmid. So this is the TI plasmid. Okay, so it will have a promoter. Say this is the promoter. Okay, and this is ampicillin resistant gene. This is ampicillin resistant gene. This is promoter, and we have a beta galactosidase gene in it. So this is beta galactosidase gene. Okay, beta galactosidase gene, and your insulin, say one produces chain A. Say this is the insulin gene which produces alpha chain. Okay, it will be introduced beside beta galactosidase galactosidase gene. Beta galactosidase gene. So this is ORI, origin of replication. This is ampicillin resistant gene. This is promoter. Okay, so promoter, all ampicillin resistant, are having a short DNA fragments lagging behind. So beta galactoside gene pakkadalli a alpha polypeptide gene will be introduced. Alpha polypeptide gene will be introduced. That you have to remember. Is that clear? Next, during recombinant insulin synthesis, bond between insulin polypeptide and galactoside can be removed by using polypeptide and galactosidase can be removed by using during recombinant insulin synthesis, the bond between during recombinant during recombinant insulin synthesis, the bond between insulin polypeptide and galactosides can be removed by using cyanogen bromide, chymotrypsin. So I'm not aware of this. Okay, so this you
Answer
can
scheme as amylase. Answer scheme as amylase.
amylase. So amylase, but it will not be digesting. I will just refer and just come back. I'm not sure of this. Prior to production of recombinant insulin, insulin obtained the insulin was in from cows and pigs were given to patient. Some of the problem faced by this treatment was loss of memory power. It reduced weight in the patient. In some human, it induced antibody production. Insulin was not active. So because of insulin introduction from cattle or swine, there was production of antibodies, okay, which is leading to autoimmune diseases. Autoimmune diseases. Next, a plant that is Rauwolfia serpentina is under the threat of extinction. To save this plant, which technique is highly useful? Genetic engineering, hybridoma, DNA fingerprinting, in vitro culture. Question number 18. Question number 18. It is Ankit. Yes, exactly. It is option 4, that is in vitro culture. So in vitro culture is nothing but, say, we can apply tissue culture. Tissue culture in order to increase production of Rauwolfia serpentina, which is also called as micro propagation. Okay. Which group of enzymes are popularly called as molecular stitches? Whether it is restriction endonuclease, ligase, RNA polymerase, or DNA polymerase. molecular stitchers. Which of the following enzymes are called as molecular stitchers? Molecular stitchers. Ligase. Lekashri, very good. It is ligase. Ligase helps in joining. We have two types of ends developed by your restriction endonuclease, isn't it? One the sticky end, ಇನ್ನೊಂದು non-sticky end, like this. Okay, so sticky end or non-sticky end, they are brought together. They are brought together by enzyme. Which is that enzyme? DNA ligase enzyme will do that work. It will join the DNA fragments together. A clone is a group of organisms produced by asexual method, and they are genetically similar. Asexual method, genetically dissimilar. Sexual method, genetically similar. Sexual method, genetically dissimilar. So what are, he is asking definition of clones. Clones are developed by asexual method, and they are genetically similar, isn't it? Genetically similar. So asexual method and genetically similar organisms are called as clone. An individual unit is called as REMET. REMET. Then match the following. Match the following. Here we have scientists and their contribution. So restriction endonuclease. Restriction endonuclease. It was given by
Sir, Purnima is asking one question.
yes.
Which enzyme acts as a molecular seizure?
Enzyme restriction endonuclease or exonucleases. They act as a seizure. Okay, because they cut the DNA. Here they have asked enzyme which act as stitcher, which will combine the DNA fragments. So ligase is a stitcher. Restriction, say endonucleases or exonucleases, they act as molecular seizures.
Thank you, sir. Thank you.
Okay. So next we have some name of the scientist. Name the scientist who gave DNA fingerprinting. Name the scientist who gave DNA fingerprinting. Okay, so DNA fingerprinting was given by Alec Jaffers. Okay, scientist who gave a restriction endonuclease. PCR was given by polymerase chain reaction was given by Carl Milles. Monoclonal antibodies. Monoclonal antibodies was given by Kohler and Milston. Okay, that means restriction endonuclease was given by Arbert. Okay, so in option ಬರ್ತಾ ಇದೆ ಇಲ್ಲಿ DNA fingerprinting was given by DNA fingerprinting was given by Alex Jaffers. So 2 are polymerase chain reaction by Carl Mills, that is 3P monoclonal antibodies, that is Kohler and Milston, whereas restriction endonuclease by Arbut. So here the option is 1. This was a bit tedious because I was not aware of Alex Jaffers um, because of some reason, and Kerry Mills for some reason. Okay, so you are aware of options now. You can make a note of it. Restriction endonuclease by Arbut, DNA fingerprinting by Alex Jeffers, then polymerase chain reaction by Carl Mullis, and monoclonal antibodies by Kohler and Milston. Some of the steps involved in gene cloning are given below. Okay, correct sequence of step involved are: first, isolation of desired gene, isn't it? First, you have to isolate the desired gene, then introduce the recombinant or introduce the isolated gene into the vector, that is the second step. Introduce the recombinant vector into the host, that is the third step. Expression of recombinant gene into the host, that is the fourth step. And extraction of recombinant gene product, that is the fifth step. Okay, so first we have to isolate the desired gene. We have to take the desired gene. So this is the desired gene. Then we have to introduce the desired gene into the plasmid. Okay, I will introduce the desired gene into the plasmid. That is the vector. First isolation, second introducing into the vector. Then you have to introduce that in the host. So this is the host. Okay. After that, the desired gene should express in that host. That means it should start producing protein. Okay, expression. So that is fourth step. Okay, third step, fourth step, expression. Then you have to isolate and purify. Okay, the gene product. These are the important steps associated with your gene cloning and its product. That is 2, 3, 1, 4, and 5. Okay, that is 2, 3. Sorry. 3, 1, 3, 1, 2, 4, and 5. That is this 3, 1, 2, 4, and 5. Is that clear? 2, 3, 1, 2, 4, and 5. So first isolation of desired gene, then insertion of isolated gene into the vector, introducing the recombinant vector into the host, expression of recombinant gene into the host, and extraction of DNA. Now, a gene from insulin has been inserted into the vector for a purpose of obtaining protein product. Such vector is called as expression vector, suppressor vector, storage vector for genomic library, none of the above. It is called as expression vector because it is going to help in development of essential proteins for our purpose. Expression vector are those which produces protein product used for genomic library. They produce protein products. Iದೇ question ಅಲ್ಲಿ ಅದು repeat ಆಗಿದೆ. E. coli is generally used as a gene cloning because it supports replication of recombinant DNA. It's easy to transform. It's free for elements and that are interference with respect to replication and recombinant. All of these. It supports replication of recombinant DNA. It is easy to transform. It is free from elements that interfere the replication and recombinant DNA. An ideal plasmid to be used for recombinant DNA technology must be minimum amount of DNA, relaxed replication control on recognition site, one recognition site for one restriction endonuclease. So it will be all the above. Okay, an ideal plasmid should be having minimum DNA, replication should be controlled, and one recognition site for restriction endonuclease. It is all the above. Then transgenic organisms are, what are transgenic organisms? They are produced by gene transfer technology, extinct organism, naturally occurring endemic species, produced by traditional plant breeding techniques. They are produced by gene transfer technology. Transgenic animals or organisms are produced by 27th question. It is one. They are produced by recombinant DNA technology or gene transfer technology. Transfer of recombinant plasmid to E. coli cells need heat treatment, UV treatment, calcium chloride treatment, or lysis. It is heat treatment. Okay, so what happens during heat treatment? There will be pores generated on the membrane through which heat shock treatment anta ಹೇಳ್ತೀವಿ. The plasmid can be easily introduced into the host by heat treatment. Okay, it is not lysis, it is not calcium treatment or calcium chloride treatment, nor UV radiation. Okay, so which of the following statement about vector is correct? All vectors are plasmid only. Plasmid phages can be used as a vector. Fungi can also be used as a vector. Cyanobacteria can also be used as a vector. Which statement is true? Whether it is 1, 2, 3, or 4. 29th question. Come on. 29th question. Bhavana, exactly. It is option 2. Why? Why option 2? Because we use plasmids as a vector, even phages as vector. Okay, we can use bacteriophage, bacteriophage as a vector. Let me tell you one thing. Okay, when we suffer from intestinal problems, say intestinal diseases. Okay, so generally we have E. coli bacteria which is growing. So we call it as coliform bacteria. And generally we observe it in our stools, feces, and that will pollute water. So when there is higher count of coliform bacteria in water, what we do is we introduce bacteriophage. Okay, bacteriophage. So what this bacteriophage will do, it will search coliform bacteria and introduce its DNA. It will search coliform bacteria, introduce its genetic material. So it is introduced its genetic material. So that bacteria will end up with lytic cycle. That means it will, the bacteriophage will kill that bacteria for multiplication. Okay, so bacteriophage will multiply. And now the coliform bacteria population comes under control. That is also called as phage therapy. Okay, we can introduce bacteriophage wherever there is higher count of E. coli in water bodies and control their population. That is called as phage therapy, and it is also used in controlling bacterial growth even in our body. So which of the following statement about plasmid is correct? Plasmids are present in bacteria, plasmids are present in all organisms, plasmids are present in bacteria and phages, plasmid are present in plants and animals. So generally we find plasmids only in prokaryotic organisms. So that is why I say that plasmids are found only in bacterial cell. Only in bacterial cell. Is that clear? Arthakta idea, Gottakta idea. Plasmids are found only in bacterial cell. Which of the following statements are not attributed to the plasmid, or they are not correct with respect to plasmid? They are circular DNA molecule. They have antibiotic resistant gene, and they have ability of autonomous replication. They have DNA that is as long as chromosomal DNA. No, this is not attribute to plasmid. Okay, so plasmid, as we all know, it is a circular DNA molecule. That is true. It is a circular DNA molecule. It will have resistant gene, that is tetracycline resistant gene, or it may be ampicillin resistant gene. They are antibiotic resistant, and it can divide or replicate on its own. So it can divide or replicate on its own. It can replicate on its own. So that is how we can say that plasmid is self-replicative. It can divide and replicate on its own. So all these statements are true, but this will be false. Okay, which of the following statement about restriction endonuclease is true? All restriction endonuclease cut DNA at specific site. This was repeated, I guess, but someone asked me what is molecular seizure? They are the restriction endonucleases or exonucleases which cut the DNA. Okay, all restriction endonuclease cut DNA at specific site, a random site, join segment, or endonucleases join DNA. It will cut DNA at specific site. So this will be the correct option. That is first one. Okay. So now tell me, which enzyme is, or which protein is identified by HIV with respect to the helper cells. Which protein will be present on helper cells, which is identified by your HIV? Can you name that protein? Name the protein which is identified by HIV on helper cells. Can you name that protein? Or receptor. Name the receptor which is present on HIV or which is present on T helper cell identified by HIV.
Student, you can type.
It is called as CD. Question is name the protein. Name the protein present on helper cell that is detected by HIV. Name the protein present on helper cell detected by HIV. That is called as CD4 receptor. Interferons are the protein produced by virus infected cells. Bhavana, well attempt. It is a good attempt. Interferons are the proteins produced by virus infected cells, right? It is not interferon, it is CD4 receptor. Remember that. You can make a note. CD4 receptor is found on T helper cell, which is detected by the spike of HIV, and it will go and bind only to those T helper cells and introduce its genetic material like that. Okay, so a small break in order to continue with the session. Now, restriction endonuclease cut DNA at a specific site, that is ligation site, or restriction sequence, or a restriction site. It is called as restriction site. Okay, so it will cut only at restriction site, which is having resistant gene associated with it. Okay. The term restriction endonuclease was postulated in 1960. The term restriction endonuclease was postulated by Arbour. You can make a note of this. Arbour was the scientist who gave restriction endonuclease name. Not Corona, not Watson, not Crick. Watson and Crick, they gave double helix structure with respect to DNA, and Corona, they explained molecular principles of replication. Okay. Then the scientist who won Nobel Prize for the discovery of restriction endonuclease are Smith, Nathans, and Arbour. Okay, Smith, Nathans, and Arbour were the scientists. Restriction endonuclease are also called as 37th question. Ankit, yes, restriction endonuclease. They are also called as molecular seizures. Molecular seizures. In restriction endonuclease, E core R1, E stands for extraction. The first letter of genus in its presence, wherever it is present, endonuclease or endangered. E core R1, E core 1, E core 1. E core 1, E stands for the first alphabet, or it stands for the first letter of the genus where it is present or it is extracted from. So E. coli, it is extracted from Escherichia coli, isn't it? So E stands for the first alphabet from which it is extracted, or where it is obtained. Okay, so first letter will represent the bacterial in which it is derived, and it's always written in capital alphabet. Okay. Then what does R stand for? R will determine restriction enzyme. R will determine restriction enzyme, and 1 will represent, that is, it will be E core Roman number 1. Roman number 1 determines which strain it is derived from. Okay. So say E core, if you say E core 1, okay, E core will represent, or E represent genus, co represent species, R represent strain, and this will represent the organism, or whether it is the same organism, same or different organism. Different organism. Say this is the first organism which was used in extraction of this enzyme. Then this divides and it multiplies and it will have different colony. So it will be considered as second antara. So now name the restriction site in which ECOR1 cleaves. ECOR1 cleaves at GAATC or CTTAAG. If you read it from 5' to 3', okay, 5' to 3', it says GAATTC. Again, if you read it from 5' to 3', GAATTC. So they are palindromic sequence. So ECOR1 cleaves the DNA at this particular palindromic sequence, that is G guanine, adenine, adenine, thymine, thymine, cytosine. Okay, so wherever if the DNA is too lengthy and if it is having GAATTC, say this side GAATTC here, and then it has a next, say it is continued, then GAATTC. ಎಲ್ಲೆಲ್ಲಿ ಈ GAATTC ಬರುತ್ತೋ ಅಲ್ಲಿ ಆ ಎಂಜೈಮ್ ಕಟ್ ಮಾಡ್ತಾ ಹೋಗುತ್ತೆ DNA ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಅನ್ನ. Okay, that is why we call it as endonuclease. Endonuclease ಅಂದ್ರೆ ಮಧ್ಯದಲ್ಲಿ ಕಟ್ ಮಾಡುತ್ತೆ ಅಂತ ಅರ್ಥ. ತುದಿಲಲ್ಲ, ಮಧ್ಯದಲ್ಲಿ ಎಲ್ಲಿ GAATTC ಸಿಗ್ತಾ ಹೋಗುತ್ತೋ ಅದನ್ನ ಕಟ್ ಮಾಡ್ತಾ ಹೋಗುತ್ತೆ. The main tools required for recombinant DNA technology: vector, desired gene, vector, desired gene, mRNA of desired gene, host, restriction enzyme, ligase enzyme. Then desired gene, host, vector, vector, desired gene, mRNA of desired gene, host. So what is the main tool required for recombinant DNA technology? Vector, desired gene, mRNA of desired gene, host, restriction enzyme, and ligase enzyme. So it is option 2, 41, option 2, 41, option 2, 40. So 40th question miss ಆಯ್ತು. Restriction endonuclease recognizes specific sequence of DNA. We call it as palindrome with rotational symmetry. Palindrome, that is option 3, right? Palindrome with rotational symmetry. Next, an example of autonomously replicating mini chromosome. Replicating mini chromosome, that is plasmid. Autonomously replicating mini chromosome will be plasmid. Which one of the following is a statement about following statement about plasmid is correct? Plasmids are mobile. Plasmids are made up of RNA and protein. Plasmid present in eukaryotic. Plasmids are present in fungi. Plasmids are mobile. Plasmids are made up of RNA and protein. Plasmids are present in eukaryotic. Plasmids are mobile. It can be carried from one organism to another, or it can be shifted from one individual to another. In some species, only the plasmid will be transported. For example, we have 6 plasmids anta barutte. So generally, F plasmids anta barutte. 6 plasmids or F plasmids. So they help in deriving conjugation tube between 2 organisms for pseudo copulation or pseudo sexual method. Then DNA ligase used in recombination DNA technology is obtained from DNA ligase used in recombination DNA technology is obtained from DNA ligase used in recombinant DNA technology is obtained from yaudado E. coli only. E. coli also ligase T4 plasmid saccharomyces. It is bacteriophage. Bacteriophage. That means ligase encoded by T4 phage. Okay, so E. coli and ligase encoded by T4 bacteriophage. So DNA ligase enzyme is extracted from, or which is used in recombinant DNA technology, is extracted from E. coli, which is ligated or coded by T4 phage. Okay. Uh, Next is DNA fingerprinting was developed by Yarado Alec Jeffrey. Alec Jeffrey was a scientist who evolved DNA fingerprinting technology. So using genetic technique in forensic science is called as genetic fingerprinting, hybridoma technology, in vitro, or gene therapy. Using techniques in forensic science, it is called as genetic fingerprinting, or it is also called as DNA fingerprinting. Okay, so genetic fingerprinting or DNA fingerprinting. Next, the technique which is called a southern blotting is used in monoclonal antibodies, genetic fingerprinting, polymerase chain reaction, in vitro culture. Okay, so here the southern blotting. Monoclonal antibodies production, in vitro culture, genetic fingerprinting. Okay, so technique that is southern blotting is followed in DNA fingerprinting, that is genetic fingerprinting. Polymerase chain reaction, it is electrophoresis, alvarez. Genetic fingerprinting is useful in identifying illegal immigrants, establishing parentage and dispute of child, identifying crimes, all of these. Okay, so genetic fingerprinting is used in identifying all these or clarifying all these features of doubts or solving these cases like criminal investigation, parental dispute, and illegal immigration. RFLP. What is RFLP? What is RFLP? That is, RFLP is commonly called as restriction, or it is elaborated as restriction fragment length polymorphism. Okay, restriction fragment length polymorphism. Okay, so which is a if a parent is having any parental disputes are there, so among people, if there is any DNA sequence that is uh, recognized by a restriction enzyme, such variation results in a different size or length of DNA, which is very common in parents or the offsprings. If this is the DNA fragment or pattern, DNA pattern from, say, parent A, and this is the DNA pattern from, say, offspring. Okay, so there are certain fragments which can be easily uh, identified by the restriction enzyme, and they are having same molecular weight or length, and they get separated at a particular distance. So ಈಗ ಇವು ಏನಾಗ್ತವೆ, they are your recognition enzymes or recognition fragments or restriction fragments. So these restriction fragments are common in, say, the parent as well as the child. So this is the parental one, ಇದು child one. Okay, so ಹಾಗೇನೆ parent ಅಲ್ಲಿ, say, mother A and father B ಅಂತ ಕೊಟ್ಟಾಗ, let us presume these are the fragments present in mother. So if it is reflected, ಇಲ್ಲಿ ರಿಫ್ಲೆಕ್ಟ್ ಆಯ್ತು ನೋಡಿ, ಅದು ಇಲ್ಲಿ ರಿಫ್ಲೆಕ್ಟ್ ಆಯ್ತು. ಸೊ ಇದನ್ನ ನಾವು ಏನಂತ ಹೇಳ್ತೀವಿ, ಇಬ್ರಿಗೆ ಈ ಚೈಲ್ಡ್ ಏನಿದೆ ಮ್ಯಾಚ್ ಆಗ್ತಾ ಇದೆ, ದೇ ಆರ್ ಮ್ಯಾಚಿಂಗ್ ಅಂತ, or their genetic character matches. So restriction fragments will be identified by restriction endonuclease enzyme, and they cut the DNA fragments with a particular length, which can be easily reflected in the child as well. And we cannot abide from this law that we have semi-conservative replication, where parental DNA will be retained constant, or one of the DNA will be retained constant. So VNTR is vulnerable nucleoside tandem repeats, vulnerable nucleotide triplet repeats. What is VNTR? Right? Vulnerable number of tandem repeats. VNTR money. Again, it comes in DNA fingerprinting, where we are going to check how many smaller fragments of genetic material is repeated in the progeny when compared to the parent. Next, 53rd question nodana. Electrophoresis technique used in DNA fingerprinting helps in separation of DNA segment, cell from DNA, tissues, RNA from DNA. What is the role of electrophoresis? Electrophoresis is a method where DNA segments are separated from the gene pool or the genomic pool, isn't it? So DNA fragments are separated. 53rd, it is 1. 54th, in DNA fingerprinting event, a small amount of DNA obtained from the sample collected at a crime place can be multiplied into millions of copies by using a technique called as polymerase chain reaction. PCR is used in multiplication of the DNA which is obtained in a smaller quotient. In DNA fingerprinting, the DNA from the gel is transferred to dash for hybridization. A DNA fragment in DNA fingerprinting, the DNA from the gel is transferred to autoradiogram for hybridization. So it will give away the image of the DNA which is separated in that agarose gel in presence of ultraviolet radiation or rays. Southern blotting is a technique used in genetic fingerprinting. It is called so because the blot is done from south side. It was developed by a scientist, E.M. Southern. It was popular in Southern America, or it was popular in southern countries. It was discovered by a scientist, E.M. Southern. That is why it is called as Southern blotting. It is a method of converting proteins, or say coating, layering protein with a dye, which makes the dye or protein visible, or the fragments of proteins visible. Okay, so Southern blotting is generally used in identifying different proteins extracted from organisms like HIV. Okay, so diagnosis of acquired immunodeficiency syndrome is done with the help of Southern blotting used in genetic fingerprinting. Then, during DNA fingerprinting, the DNA nucleotide hybridization was with a probe can be detected through DNA nucleotide hybridized or DNA nucleotides hybridized with a probe can be detected through electrophoresis, polymerase chain reaction, hybridoma, that is auto radiography. During DNA fingerprinting, the DNA nucleotide hybridized with the probe can be detected through okay, it is called as audio radiography, which is similar to X-rays. but it will be done with the help of ultraviolet radiation. So there will be a slab like this, that is your agarose gel. Okay, and they illuminate this agarose gel with the help of ultraviolet radiation. For example, this is agarose gel. So after illumination, your DNA fragments will appear something like this. They appear something like this. They will get illuminated. Okay, and you, based on their distance or based on the association, we can count. So you can say that this is lighter and this is heavier. Okay, so that illumination is called as auto radiography. DNA fingerprinting, DNA nucleotide which is hybridized by a probe, it will be detected by auto radiography. Either repeated, it is all of these. So RFLP, VNTR probes are some of the technology associated with DNA fingerprinting. Okay. Then Dolly, the first animal produced through cloning, is camel, rat, cow, or sheep. 60th. Who is Dolly? Whether it is a camel, yes, Mona Lisa. It is a sheep. Okay. So the clone which was derived, that is Dolly, by somatic hybridization or in vitro hybridization, was a sheep. So some of the steps involved in DNA fingerprinting are listed. Okay, so whether collection of the sample, extraction of DNA, treating DNA with restriction endonuclease, gel electrophoresis, transfer of DNA into nitrocellulose, autoradiograph. So it is 2, 1, 3, 5, sorry, 3, 6, 5, 4, and 7. It ಬರುತ್ತೆ. 2, 1, 3, this is 4, 4, 5, 6, and 7. So first collection of the sample, then collection of sample, then extraction of DNA, then you have to treat it with restriction endonuclease, then gel electrophoresis, transfer of that in nitrocellulose membrane, then hybridization and autoradiograph. A person is suffering from atherosclerosis. Since conducting a surgery is very difficult due to his age, which of the therapy can be advised to him to overcome the problem? Gene therapy by anticoagulates, gene therapy for formation of anticogenic factors, the genetics and DNA fingerprinting. So here both A and B are can be followed in order to overcome atherosclerosis. Okay, so which is again a kind of genetic disease. The gene therapy can be made highly effective by conducting gene therapy for each and every tissue, gene therapy through injecting modified cells, gene therapy for stem cells and bone marrows. Okay, so gene therapy can be modified or it can be made highly effective by including stem cells and bone marrow cells. Gene therapy helps in curing genetic diseases. In 1990, the first gene therapy was conducted, that is SCID. Okay, it is not cancer, it is not AIDS or malaria. Okay, so what is SCID? That is severe combined immunodeficiency disease. Okay, next, during gene therapy, the possible way through which the genes can be introduced in the cell: micro injection, small viruses, both 1 and 2, erythrocytes. It is both 1 and 2. Okay, so micro injection is a method where directly the genetic material or vector or a plasmid will be introduced into the host body, or we can use small viruses like retroviruses, like HIV, non-infective HIV can be used. The one of the type of gene therapy, that is functional genes, are introduced in sperm or the egg. This is called as somatic cell gene therapy, germline cell or gene therapy, gametic gene therapy, vegetative cell gene therapy. Okay, so gametic, it is functional genes are introduced in sperm. When functional genes are introduced in sperm, it is called as germline gene therapy. Okay, what is germline gene therapy? It is a method where functional genes which are required, for example, a DNA segment is required for production of a specific protein. Okay, they can directly introduce it in the gametes. So, for example, this is egg. Eggly, a portion of DNA can be replaced, and they can introduce that DNA into it, and finally it can be used in fertilization. So this portion will be expressed even after fertilization. That is called as your germline gene therapy. The somatic cell gene therapy, the functional genes can be introduced into sperm, antibody cells, germline cells. Somatic cell gene therapy, antibody cells. So gene can be introduced into antibody cells. Next, 76th question nodi. Okay, hope you are with me. 76th question. 76th question, if you take BT cotton, a cotton variety obtained by crossing two different cotton plants, a cotton variety brought from South America, insecticide sprayed on cotton plant, transgenic cotton variety. What is BT cotton? What is BT cotton? It is a transgenic, exactly. It is a transgenic variety of cotton. In biotechnology, mass culturing of cell, microbes, mass culturing of cell and microbes are achieved by using test tube culture, bioreactive, autoclave, electrophoresis. It is bioreactor. Bioreactors are the huge vessels, okay, which can be either sparge tank or it may be stirred tank, bioreactor. Okay, so bioreactors are the mass culturing vessels used in culturing microorganisms and other produces. A device in which substrate of low value is utilized by living cells of organisms, so to generate or produce higher value compound, that is bioreactors. ಇಲ್ಲಿ ಎರಡಕ್ಕೂ ಬಯೋರಿಯಾಕ್ಟರ್ ಆಪ್ಷನ್ ಬರುತ್ತೆ. ಬಯೋರಿಯಾಕ್ಟರ್ ದೊಡ್ಡ ದೊಡ್ಡ ವೆಸಲ್ಸ್, ಅದರಲ್ಲಿ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ಸ್ ಹಾಕ್ತಾರೆ and they use nutrient medium and this nutrient medium is consumed and we get larger concentration of individuals or higher concentration of individuals. Okay, so 86th question, if you take totipotency. What is totipotency? It is ability of plant to develop into a complete plant or ability of a plant cell to develop into a complete plant. That is called as totipotency. Somatic embryos developed from egg without fertilization are called as, or embryo-like structures developed from cells of calyx. Okay, so generally when we place a single cell in a nutrient medium, it will divide and produce a cluster of cell. This cluster of cell is called as callus. Okay, so this callus further differentiate into embryos. So we call it as heart-shaped embryo, or say globular embryo, or torpedo-shaped embryos. Okay, so later these embryos will have embryonal axis developing into plumule as well as radicle. Okay, so that is, somatic embryos are developed from structures of callus. Okay, so golden rice, hybrid rice developed by traditional breeding, rice variety developed by plant tissue culture, a rice variety obtained by rDNA technology, hybrid rice developed in rDNA. It is a traditional breeding program. Rice variety developed from your traditional breeding or plant breeding method. So golden rice was developed by Okay, so the golden rice was developed by Ingo Forticus and Peter Bayer. Okay, so these were the scientists who worked on golden rice variety. So gene required for transferring rice plant into a golden rice was obtained from gene required for transforming rice into a golden rice was obtained from. Okay, it was obtained from carrot because beta carotene. Yes. Okay, so there are many questions apart from this. I will be sharing you this PDF format. Okay, simultaneously go through it, solve as many as MCQs. It will help you a lot in coming days. Okay, so with this note,
Yes, yes, sir.
all the very best.
I think Abhishek, sir.
Yeah.
Thank you so much, sir.
Yeah, thank you.
Yes, dear students, is beautifully explained, and our Dayanand Sagar University's motto is such a experienced faculties are reaching up to you, because in your area it is not possible to bring them. It is online class has been conducted. Hope all of you are enjoying. Thank you so much, Abhishek sir. It's
Yeah,
beautifully explained, and many questions you have taught, sir. Thank you so much for that.
yeah, thank you. So one question I just wanted to answer. Someone asked, Bhavana asked, atherosclerosis. Atherosclerosis, it is a development of cholesterol in our body, which will develop clots, or which will block the arteries or vessels or blood vessels by deposition of fat on artery walls, and it will lead to cardiac diseases. So that is atherosclerosis, and that can be treated by genetic engineerly introduced factors which reduces enzyme developing uh, triglycerides. So we produce triglycerides, and these triglycerides is because of oil what we consume, and they go and attach to the vessels, that is arteries and veins, and the enzyme will be more active during night. Daytime the enzyme is not synthesized, and in night the enzyme which builds cholesterol is synthesized. That is why we say that you should consume low cholesterol meal or low fat meal during night, because in night the food whatever we consume is directly digested, distributed, and cholesterol is being constructed. So if a person is aged and he cannot undergo a bypass surgery or an open heart surgery, or it may be uh, uh, angioplasty, they can introduce enzymes that can digest those clots or cholesterol lowering agents. For example, statin is very best. Statin tablets are available. We can introduce statin in the form of injection, and we can avoid the growth of fat in uh, or accumulation of fat in that body. Okay, so hope you have enjoyed the session. We will continue in the coming sessions. Till then, take care, enjoy reading, enjoy exploring. Take care. Bye-bye. Thank you.
Thank you, sir. Thank you so much. Dear students, feedback you can share on helpline number. You can WhatsApp. Thank you. Thank you all.